Why don't you tell me? Use a word to describe this response to me. Someone, give me a word. Right? Just use one word. Doesn't matter. Someone? Can you use one word to describe the secondary response to me? Come on. 用一個字去形容啊。Peak. Okay, peak. Okay, that's good. So there's a peak here. But this one has a peak too. It's a lower peak though, right? 有冇第二個字去形容呢個 ？Stronger. That's good. That's a nice word. Stronger. And faster. That's good. Exactly like this.
Work it harder, make it better, do it faster, makes us stronger, more than ever. After hour, work is never over. Work it harder, make it better, do it faster, makes us stronger, more than ever. Hour after hour, work is never over. Work it harder, make it better, do it faster, makes us stronger, more than ever. Hour after hour, work is never over.
That's a great song from the 90s, okay? That from Daft Punk, right? Harder, better, faster, stronger. That's what the secondary immune response is about, right? This for the adaptive though. This is adaptive, right? Keep in mind, right? This is just the adaptive immune system, but an innate response never changes, never changes. But there is a catch though. There is a catch to this, right? What is the catch? Well, take a look here. If we focus a little bit here, right, the mRNA vaccine will only work if the viral protein gets successfully translated, okay? Only if it gets translated into a viral protein that you will give rise to all these benefits, okay? Unfortunately, our cells actually do have natural, innate antiviral mechanism to suppress this, to inhibit translations of viral RNA, okay? So if we take a look at this endosome right here and magnify it bigger, this is what you see, right? You see that inside there are these question marks right here. What the heck are these question marks? Well, they are called toll-like receptors, TLRs. They are just like policemen that are standing inside this endosome, and they're constantly checking whether there are some sort of viral nucleic acids that are floating around. And once they see some sort of viral nucleic acids around, then that will lead to the activations of these receptors, and there will be release of what we call interferons, IFNs. And now I'm getting confused. What are these IFNs? Well, if you have a police, you found something, what do? You make a big fuss about it's a siren. It's a kind of antiviral signal rapidly and broadly modulating the innate immune system, right? It's a big siren to ask for help, right? It will induce nearby cells to suppress translations of proteins and sometimes even get them to carry out cell death, to kill themselves, right? So that there will no longer be any viral protein being produced. Now, keep in mind that this is actually part of the innate defense, innate immune system. It has no memory, okay? So despite the facts that this is wonderful, right? This is actually triggering an immune response. We want our vaccines to give us response, right? But this is innate, so it does not help at all. Because next time when we face the COVID-19 vaccine, it will not make us harder, better, faster, stronger, just like that song, okay? So, and also, most importantly, IFN. It has an effect of increasing expressions of PKR, okay, PKR. And what is this PKR right here? This is protein kinase R, right? It's an RNA-activated kinase. And as just its name is suggesting, it's activated by viral RNA. When the cells experience this sort of well, usually the expressions of PKR is rather low, but when the cells experience this sort of viral nucleic acids and this sort of endosomal TLRs are activated, and then cells will start secreting IFNs, like the big siren is now on, right? These will actually increase the expressions of PKR. And what does PKR do? It's a kinase. So what it does now, this increase the PKR, will now phosphorylate this sort of translation initiation factor 2 alpha, EIF2 alpha, eukaryotic initiation factor 2 alpha. Okay. And once it phosphorylates this protein will then do something really terrible, which is globally inhibit translations of all mRNA and promote apoptosis. Normally, if there is a real virus infecting you, this would be great because this is an antiviral mechanism so that there won't be further translations of real viruses, right? But now, in this case here, it's not that great because it significantly reduces the effectiveness of our mRNA vaccine, right? So now, what we want to do is we don't want to activate the PKR at all. Take a look here. This is a comparison of vaccine efficacy, right? Previously, there is a company called CureVac, and they produce mRNA vaccines, and the translation efficiency is only 48%. Okay, not that great. But if we take a look at BioNTech and Moderna, these companies' mRNA vaccines goes above 90%. Wow, that's much more impressive, right? Well, are they using different kinds of proteins for the mRNA vaccine? No, both of them uses the same S protein of SARS-CoV-2. And both of them uses the same kind of lipid nanoparticle to deliver them as well. So why? What's the difference here, right? The difference is that CureVac uses unmodified mRNA, aka uridine, right? Whereas by BioNTech and Moderna, they uses modified RNA with pseudouridine. So what is this pseudouridine here, right? I don't even know what uridine is, okay? Right, what is pseudouridine then? Well, pseudouridine is an isomer of the nucleoside uridine, right? U in RNA, right? In our DNA, of course, we have nucleotides, right? We have the A, C, G, T bases, right? Remember that, right? And then for RNA, we don't use T, right? We use U. So here, this is an unmodified mRNA. You can see how C, G, A, G, U, U, G, C, right? C, G, right? Okay. And the for pseudouridine is the most abundant post-transcriptional RNA modification in cells to regulate gene expressions. So this is a modified one where all these U's will become modified to become pseudouridine. So this is the pseudouridine modified mRNA. Now, in terms of molecular structure, this is the uridine, right? The U in here, right? So this is the, so instead of the usual C1N1 bond in the uridine, this is the, let me press here, right? This is the uracil. This is what's called the uracil. And this is the ribose sugar here, right? Instead of the usual C1N1 bond in the uridine, this is the C1 here. And this is the N1 bond, right, in uridine. The pseudouridine has a CC bond between the C1 of the ribose sugar and C5 of uracil, right? So this is, if you turn this mirror flip, right, like this, and flip it to the other side, then you get this part right here, right? You see how the N1 goes to this side, comes here, and there's actually a C here that will be flipped onto this side. Now the C will bond to this C right here. So this is the C5 of the uracil, which is bonding to the C1 of the ribose sugar. So instead of the C1N1, now you get a C1, C5 bonding, the CC bond, right? CC bond right here. That's in the pseudouridine. So if we take a look at the history, right, of the pseudouridine research, it can go way back, right? It goes back to like almost, 1869, right? That was really long ago, right? But at that time, you know, there are a lot of interesting findings, but nobody has ever thought of the actual application of this, right, in mRNA vaccine. It's only until all along this pathway, right, all along these different researchers by different scientists, right, finding that, you know, pseudouridine actually naturally exists in our cells, what the functions are, and until finally here, in 2008, right, Dr. Carrico, right, has a paper in molecular therapy that talks about how immunotherapeutic studies with this sort of pseudouridine in mRNA, that finally people realize the real applications of pseudouridine. So here this is the paper in 2008. So this is Professor Kathleen Carrico and also Professor Drew Weissman, right? So they have this paper, you can see the title, Incorporation of Pseudouridine into mRNA, yields superior non-immunogenic vector with increased translational capacity and biological stability, right? Non-immunogenic, right? So it means does not trigger as much immune response, okay? So you're like, well, but it's a vaccine, right? You want to do this in vaccine. Shouldn't we trigger immune response, right? So that it can actually do better? Like I said before, yes and no. We want to trigger the adaptive response, right? We talked about that, the cell-mediated response and humoral response, but not the innate response, because that doesn't help at all, right? Then that would only result in a lot of very severe symptoms, right? In a severe COVID-19 case, right? But it has no memory of it. Next time it's going to do exactly the same thing, right? Okay, so this discovery really changed the world because of this, right? There is a way for us to effectively translate these mRNA into proteins, practically turning this sort of mRNA from 48% to 90-something% effectiveness in translation, right? For this, and this is why, you know, just having the discoveries in the past is not enough. Just like if you know about the, you know, CRISPR, right, editing, right? The original discovery of CRISPR was a long time ago, but it was until, you know, Jennifer Doudna that found the real application of gene editing that actually managed to put it into the spotlight. Here, just like this case here, discovery of pseudouridine was a long time ago, but it was until these scientists that put them into real application that managed to save millions of lives that's actually much more meaningful, right? So that's why these scientists managed to win the Nobel Prize. So if we take a look at pseudouridine here, right? So psi can be found in many types of RNAs, not just mRNA, right? Transfer RNAs, ribosomal RNAs, messenger RNAs, and even small nuclear RNAs, right? All these different kinds of RNA in us contain pseudouridine. And also you can find them at home as well. You can see me, I'm just holding this psi right there. Turns out that this is, of course, this is just for combing my daughter's hair. But yeah, you can find these sort of psis everywhere. Anyway, so this is just for fun. So, okay. So potential functions of psi, right? It can strengthen the RNA-RNA interactions and RNA-protein interactions, right? Via additional bonds with neighboring atoms. It can promote RNA stability, right? So that it can increase the lifetime of mRNA. It can promote translations, right? So it increases the translational efficiency. And lastly, also prevents immune responses. It prevents the production and release of IFNs. So reduce the amounts of innate immune responses in us. And that's very important, right? So this is the effects of pseudouridine, right, on PKR. For viral mRNAs, they don't have pseudouridine. For viruses, they always use uridine, right? And these actually strongly activate PKR. So there will be global inhibition of translations of all mRNA. Whereas pseudouridine modified mRNAs, they sometimes do still activate PKR, but to a much lesser extent, right? Much lesser degree. So it allows much more effective translations of the vaccine mRNA. And this explains why, you know, BioNTech and Moderna's vaccines are much better efficacy than those from CureVac. So why did CureVac choose the unmodified mRNAs though? Well, perhaps they thought it would be better to mimic the actual SARS-CoV-2 virus, right? Because the actual virus uses unmodified mRNA, right? So because usually better mimic equals better results, right? If we can mimic the virus better, right, perhaps we can get a better result. So that's perhaps one of the reasons why they did this. But maybe that was not the case here. Well, why did the SARS-CoV-2 virus not run into the same problem as the CureVac mRNA vaccine of activating PKR and inhibiting translation, right? If the CureVac mRNA inhibits the translations and end up having a bad efficacy of translation, shouldn't SARS-CoV-2 having the same problem, right? So we shouldn't get infected so badly, right? But that's not the case, right? So why? Well, it's only just this year that some new findings came out. SARS-CoV-2 virus actually has some sort of nucleocapsid protein that can inhibit the PKR-mediated inhibition of translation, basically. Okay, so now we found that, oh, so they thought they were mimicking the virus, but they only mimicked half, right? Only mimicked the mRNA, but they didn't mimic on the sense that the real thing actually has a de-weaponizing weapon to actually get rid of our innate defense. But if you just inject the uridine, right, unmodified mRNA, it doesn't have that function. So they would be completely fallen under our protections of the innate antiviral defense. So the future, right, of our mRNA vaccine, right, of course, our war with COVID-19 has been devastating, right? But on the bright side, it has facilitated us to develop this new weapon, mRNA vaccine technology, which can now be used to combat many other diseases, including influenza, Zika virus, right, deadly viruses, rabies, or even cancer in the future. We may be able to use this new technology for our advantage, okay, to our advantage. And here, pseudouridine plays a very important role that allows all the mRNA vaccine to be effective. And so now this becomes highly efficacy in inducing adaptive immune response, right? Adaptive, right? High efficacy inducing adaptive, but low in inducing innate, right? Easy of design and modification. Making an mRNA is much easier, right, than actually making like inactivated viruses, right? And much higher specificity too. And there's a low cost in manufacture as well. So we can expect there will be rapid development to continue with mRNA vaccines in the near future that will protect us and improve the lives of everyone. Hence, this you and I really changed you and I. So that's the end of my talk. Thank you very much.